Hello, this is the last of the 3 videos for Biology 30 Final Exam Review. This one is going to be talking about Unit 6. In Unit 6, we looked at, well, a lot of things with to do with DNA. And one of the things we talked about towards the end of the unit was the Hardy-Weinberg equilibrium. And that's something that we learned about. We said that this applies and has two formulas, P plus Q equals 1 and P squared plus 2PQ plus Q squared equals 1. Uh, and that applies only sometimes when there is no random, selection, nothing that is making one genotype more advantageous than another, no big mutations going on, no net migration, there's no big population shifts. There has to be a large enough population that you can do statistics on it, and mating has to be completely at random. We also said that this is not very realistic and that most populations in the wild are not at Hardy-Weinberg equilibrium. But it's a good baseline. So Hardy-Weinberg equilibrium had this two sets of equations, P plus Q is 1, or P squared plus 2PQ plus Q squared, get out of there, equals 1. And in this case, our P was the dominant allele, our Q was the recessive allele, and this P squared was when we had a P and a P. This would be our homozygous dominant. For Q squared, this would be our homozygous recessive. And for 2PQ, this represents in our Punnett square this guy and this guy. So if this is our homozygous dominant and our homozygous recessive, depending how we set it up, these two are always going to be the same. And there's two of them, that's why. And this is our heterozygous. So it has a mix of the dominant and the recessive. So maybe like a big B and a little b. Whereas this one would be two big B's. And this one would be maybe two little b's. This is telling us that if we add up all of the alleles, the dominants and the recessives, since that's all we're looking at, that's got to be equal to 100%. And this is telling us that if we add up all of the homozygous dominant individuals, the homozygous recessive individuals, and the heterozygous individuals, that's also got to equal 100%. Here we say if the Hardy-Weinberg equilibrium predicts that an allele frequency should be 25%, so 0.25, we usually do this in decimal form, but it's observed to be 35%, so higher than expected. The phenotype controlled by that allele, must have some kind of advantage. So it would not fall under Hardy-Weinberg because the prediction didn't work. And since it's happening more in nature, we're seeing it surviving more, higher than we expect it to be. We expect it to be 25%, but it's 35%. Then it must be somehow advantageous. If this number was 15% instead of 25%, then that would be a disadvantage because it's showing up less than we would expect. Going to move over to the right here. We're going to talk about some DNA stuff. And one of the ways that we can work to fight infection is with antibiotics, and they can work in a lot of different ways. These are some things that antibiotics really could do, and one of them is that it can prevent mRNA from joining with ribosomes. If it's preventing mRNA from joining with ribosomes, then what's going to happen? Will it make any proteins? Will it make the wrong proteins? Because that's what the point of DNA is, right? To encode for proteins. So if mRNA is not joining with the ribosomes, then we should get nothing formed because the ribosome is the structure where the tRNA brings the amino acids in to join up into the polypeptide chain. And if we're not even getting there, then we can't start a polypeptide chain, so we don't even get a tiny protein. If it inhibits the formation of peptide bonds, again, the whole point is to join one amino acid to another amino acid to another amino acid to another until we get a big polypeptide chain that we can fold up into a protein. And if we can't make those bonds, then we just have a whole bunch of loose amino acids floating around, and that doesn't help anybody. If an antibiotic causes misreading of the mRNA code, so it causes the letters to get garbled up, in that case, we might get a protein formed. It might be shorter than normal because you might have a stop codon by accident. It might just be totally the wrong amino acids and you end up with a useless protein. But you might be able to build something out of it. If an antibiotic prevents one particular amino acid from being added to a polypeptide chain, so say it just doesn't like leucine, I don't know why, then where we have one being added to the next, being added to the next, and then suddenly this one can't go in because that's its least favorite one, then we end up with something, a short polypeptide chain. Maybe we could call it a short protein if we were being generous, but it would not be as long as possible because whenever that amino acid does show up, then it won't be able to plug in. It won't be able to join on. And then here we can't have a gap. So that's just the end. That's the end of that thing, shorter than it should be. If an antibiotic prevents nucleotides in mRNA from sticking together, well, you don't get mRNA then, right? If you don't have the nucleotides, so the guanine, cytosine, adenine, and uracil, because it's RNA, not thymine, then we can't make that mRNA, which means we can't have the mRNA leave the nucleus and go find the ribosome and go start this protein building process. So we end up with, again, no proteins. If an antibiotic prevents tRNA, so transfer RNA carries these little amino acids to meet the ribosome to join onto the polypeptide chain. If it prevents tRNA from binding to a start codon, we get, again, no proteins forming. So antibiotics can work in a lot of different ways, but here we can see a bunch of the ways involve DNA in the way that we can say they either prevent that pathogen from creating the proteins it needs to infect you further, or have it make the wrong or a broken protein, which again means it can't do its job. When we were looking at mRNA and tRNA and the process of transcription, translation, we had the idea of complementary triplets. So a complementary triplet for G, A, T, G is always paired with C, A is always paired with T, and T is always paired with A. C and G and A and T are always pairs together on DNA. When we are going over to RNA, when we're making RNA, then we have the same guys, GAT. But, okay, G pairs with C, that's fine. A pairs with T, but in RNA, T doesn't exist. Instead, we have uracil. And T pairs with A, and that exists, that's fine. So the complementary DNA triplet of GAT would be CTA, and the complementary RNA triplet would be similar, but instead of T, we can't write T in RNA, so we write uracil, we write U. And that's related to this idea over here too. Can a specific amino acid be represented by only one codon? And you just have to look at your data sheet. And your data sheet is there on your exam. I would recommend printing out a copy to have just in front of you because it's quicker. But we can see that we've got 2 to make phenylalanine, or 2, 4, 6 to make leucine, or 4 to make isoleucine. So no matter what we look at here, except for if we look at methionine, all of these other ones have at least 2 ways to make them. So the answer here is yes. You could have UUA or UUG. The A or the G here could be different to make leucine, and you'd still get leucine. So if we come back over to here, which substitutions would cause a silent mutation? ACG to UCG. We have to look at our chart to find this out. ACG is going to be, we look at A for our first nucleotide, C for our second nucleotide, and G down here, G, we get threonine. So this is threonine. And UCG, so UCG, is serine. That's not silent. That's a missense mutation. So not that one. That one is no good. ACU to ACC. Here's A. I'll just get rid of my circles from before. ACU. That's threonine again. And to ACC, that's also threonine. So these two both encode for the same amino acid, threonine, which means that they make a silent mutation. So this is our one that gives a silent mutation. You should always check the other ones as well, but I'm trying to keep the video as short as possible for you, and they do end up with different amino acids. When we're looking at the types of mutation we know can be silent, missense, or nonsense. Substitution is most likely to result in silent or missense, but insertions and deletions are the ones that are more likely to result in nonsense mutations. This I actually meant to have up here when we talked about DNA and RNA triplets, the idea that if we have a match on a DNA section to an RNA section, if this is a C, and all of the ones that are shaped like that are Cs, this pointy one here, that's a C. If these are all Cs, then they would pair with Gs on DNA and also on RNA. But if we look at the one that's different, and the different one is, well, they all have the same shape in this picture, but C's pair with G's, A's pair with T's, except for on RNA, where an A will pair with uracil. In the DNA molecule, we have our sugar, our phosphate, and our base to form a nucleotide. Over here, to make large quantities of a DNA fragment, the enzyme that you would need would be DNA polymerase. You can call it polymerase, or to be better, you could call it DNA polymerase. To glue strands of DNA together, you need the gluing enzyme, ligase. This also works to pair bits of your Okazaki fragments together. To unwind a DNA double strand, to unwind that helix, you use the enzyme helicase. So just remember that these enzymes are important. They have jobs to do in both translation, transcription, and DNA duplication, replication. Separating strands of DNA by their weight and charge. This is done with a process called gel electrophoresis. And so gel electrophoresis, I have a little animation to make bigger for you here. Gel electrophoresis is a good way to find out things like, does the DNA sample match the crime scene, or is this baby guppy really the offspring of that mom and that dad? And for gel electrophoresis, you chop the DNA sections into bits, and you put them on a charged plate. They're swimming in a gel, and the heavier ones are going to stick closer to the beginning, and the lighter, smaller ones are going to make it to the end. And they'll be separated reliably by size and charge. And you'll find that the baby guppy has some bits that match up with their father's DNA, some that match up with the mother's, and none that are unaccounted for, because they all had to come from somewhere. This is here just to remind us of the structure of a nucleotide, phosphates, pentose sugar, whether this is RNA or DNA, and the nitrogenous base, which is either C, G, A, T, or for RNA, the T would be replaced with U. The bonds between two pairs in the double helix of DNA are hydrogen bonds, and those are the ones that are broken by the enzyme helicase. When we are doing DNA replication and transcription, translation, these are processes that you should definitely study. For replication, we have to unzip the strand with helicase. We have to take polymerase to build in our replacement or our secondary strand. And on the leading strand, this goes nice and smoothly, but on the lagging strand, we're going in the wrong direction. and that results in those sections called Okazaki fragments that need to be glued together with the enzyme ligase. For protein synthesis, which is transcription and translation, we know that the mRNA so we have to unzip the DNA molecule with helicase. We have to get to the nucleotides. We have to get to those bases and read them. We make a copy on RNA called mRNA. That mRNA is going to leave the nucleus. It's going to find a ribosome. And once it's at the ribosome, then the tRNA is going to bring over one amino acid at a time that's going to be matched into place. That builds, as we add on those amino acids one by one by one, that builds into a big polypeptide strand, which eventually will become a protein once we fold it up. The last thing I'm going to mention, we're over 16 minutes here, is for populations, one thing to remain aware of is different types of strategies, including K versus R selection. K-type species are ones like us. We don't have that many babies. We put a lot of effort into them. We hope that they all survive. We have smaller babies than we are. We take care of them. Whereas in our selected species, they have a lot of babies really quickly, and a lot of them are the same size or ability as the parents, and they take less care of them. So many, many, many small, sorry, uncared for offspring, or taking the time to have fewer offspring, but caring for them more. Those are the two different strategies. So this one was a bit longer. Maybe next time I should break it down into two. But these are 3 videos to help you study before your final exam for Bio 30. If you have any questions, you know where to find me.